have like other things like that. Like, can you take like half out kind of thing? If we can let you know about that, let's say you have it like 10 in the morning, but then come afternoon.
So do you have to pour any time?
But process like let you know about this in that.
Yes, please. But at least I expect you to attend half of the day.
Oh, yeah, I will be.
We will take group pictures. So please make sure you are here. Tomorrow, no classes anyway, so that's why I was not expecting you. Yeah. All right, go ahead.
Hello, my name is Grigore. I am a second-year PhD student in bioinformatics, and my review paper is on whole genome alignment. What is whole genome alignment? So it's when we have two genomes, A and B, and we need to find a mapping from each position in A to its corresponding position in B. So that sounds simple when the genomes have the same length, but it becomes really challenging when we have different rearrangements. So consider this example when we have two mismatches, three gaps, and these four positions are matching in reverse complement. So then the problem becomes very non-trivial.
What do you mean by reverse complement?
So this is, if this is G, then this will be the corresponding nucleotide, which is C, and it's reversed. So this was the last character, so it becomes the first, and it becomes the corresponding nucleotide. So G and C and A and T. So G and C, and then AT, CG, and AT. So then there is another term, global alignment, which sounds kind of similar. So I wanted to point out what is the difference between whole genome alignment and global alignment. So the main difference is that when we say that we align entire genome, we don't necessarily imply that the entire genome consists of only one sequence. Entire genome can be consisting of many contigs, like shorter fragments of a genome. But for global alignment, we consider like two individual sequences, and we try to find the best start to end alignment. And also for the whole genome alignment, we try to find rearrangement. So yeah, for global alignment, we just focus on mismatches and gaps, and the algorithms will be different for these two. And speaking of the algorithms, these are the tools that I considered for my review paper. So there are two key approaches, seed chain align and seed and extend. So these all follow seed chain align algorithm, and seed and extend is used in BLAST tool. So BLAST is not listed here, but we used it for another project. And basically I am comparing all these tools to find the best tool to output the total number of insertions, deletions, matches, mismatches in two sequences. And so these tools differ significantly in terms of input and output formats. And most of their output formats are not human readable. So they are like, if we open them as text files, they will be just like a bunch of numbers, like coordinates for individual alignments. And yeah, so the only one that was kind of readable is what it's .report, because it contains the total number of SNPs, indels, and so on. And so for the two of these tools, they require something else besides the two sequences. So progressive cactus, apparently it is named progressive because it requires a progressive tree, which is another term for phylogenetic tree, and it aligns sequences in a certain order. So basically this tool is for evolutionary biology, and another tool requires some external files like scoring matrices. So it cannot just take two files and give an alignment. Yeah, and so the algorithms differ significantly, but all of these will require like a separate presentation to explain, because they all have like a different key idea. But as I said, so these all share the seed chain align algorithm, and the other algorithm is seed and extend, which is used in BLAST, and I'll show you the seed and extend heuristic, which is used in BLAST, and it's used very successfully. So the heuristic is very simple. You take the query, you break it into words. So you take first 3 characters, then second 3 characters, and so on. And then you make a table of similar words like this. These are different by 1 character. This is also different by 1 character, but it's in the middle. And so then you take this table and you find exact match between the table word and the subject sequence. And then you extend the match in both directions, and then you allow gaps, like you make gap extension. And this is how we get high scoring pairs in BLAST. And so the output will be, the output in BLAST will be just like a bunch of high scoring pairs, like individual alignments between separate regions of 2 genomes, and there will be many of them. And as I said before, it can be 2 sets of contigs, and then we can observe that one region of contig number 1 matches to a random region from contig number 25, and it also matches to a random region in contig number 2. So that's whole genome alignment. So when we talk about evaluating bioinformatics tools, it makes sense to focus on runtime or memory usage. But in my review paper, I try to assess them based on how well they can quantify the number of matches, mismatches, and indels. And the reason why I do that is because I have another project where we need to run the tool to compare 2 bacterial genomes, and we want to get the overall genome similarity. And to do that, we just try to count the overall number of matches, mismatches, indels, and then when we know that, we can derive the overall similarity and try to apply different formulas with using these numbers. And so I took 2 examples. So the first example is a real example of 2 genomes, 2 complete assemblies of this species. They come from real bacterial reference databases, RefSeq and Patrick. And another example is artificial. So the artificial example I made myself. So this is the first example. And this image is produced by a tool, like software, that took a BLAST output and made this image out of this output. So in this image, this white rectangle is the subject, because it's RefSeq, and we align Patrick to RefSeq. And this will be the query. And this is like a legend. It shows how this blue rectangle aligns to the white rectangle, and so this and the gradient shows the direction of the alignment. So it goes from more transparent to less transparent. It means forward direction. If it goes the other way, then it's reverse complement. And so from this visualization, you can see that there is this gap. And basically, this image I made myself. And just to illustrate this gap, it's at this position, 1, 2, 1, 4. And so I used this real example first to try it with every tool. Try to align these two genomes. And so I couldn't use these two because they required something else besides the genomes. They required external scoring matrices and some soft masked input and phylogenetic tree, which I don't have. But for these two, for Mummer and GS Align, I was able to generate some results. So Mummer outputs the percentages for query and subject. And so this becomes like 93 and 97 for it gives like percentage identities separately for query and subject. And then I got this many SNPs and indels. And for GS Align, I got a different number of SNPs, which is basically illustrates the challenge, because when we take a real example, we don't have the ground truth, and so we get different results from different tools. And so to address this limitation, I took an artificial example where I know exactly how many indels and mismatches and matches I have. So this example consists of 4 distinct regions. The first region from 0 to 500 base pairs is a complete match. The second region from 500 to 1000 is a complete mismatch. And then from 1000 to 1500, we have a match, but it has 5 indels. And then from 1500 to 1701, we have 21 mismatch. And so the total length for the first sequence is 1701, and for the second sequence is 1700, because here we have 2 and here we have 3 deletions. And so BLAST was able to capture all of it very accurately. And so, yeah, this is what I got with these examples. So the number of sequences is the same. The total sequence length is the same. Then aligned bases, Mummer shows for both query and subject, but for the queries, it's the same. Then unaligned bases, for the query, it's the same, but Mummer has also 504 for the subject. And then the number of SNPs. So I got 21 SNP from Mummer, which is exactly what I expected to get. But from the other one, I got 233, which doesn't make much sense.
What is the
Single nucleotide variant. That's how they call it.
Okay.
Yeah. Because I copy pasted from the output itself, from the output of the tool. So that's why it's different, but yeah, it's these are SNPs. And so then I got a different number of insertions and a different number of deletions. And so the average identity, it makes more sense for the GS Align, actually. But in this case, it might be, Mummer might output the percentage identity only for reference or query. like the number of matching positions divided by the total number of positions. Yeah. Yeah, and also, yeah, that makes sense because here we have the total alignment length of 1740, and it gives the identity for the 1740, and in here it gives it separately. So this number is probably for the reference.
So the higher the best for the identity.
I mean, for the identity, I mean, yeah, the general idea is that the higher identity would have the better, but in this case we have like a different length. So I considered that Mummer 4 was better because it was able to capture 21 mismatch.
I
Yeah.
like that. So you understand what was happening. So compare two tools with no ground truth to tell whether this is correct or not correct. So what he did, posed his own errors. We know where are those errors and how many. And he ran two tools and challenging the tools to figure out or discover those errors that he already imposed himself. And one of the tools predict them correctly, the other one just nowhere showed 200 errors, which is only 21 errors.
Yeah.
But for the other two tools, couldn't find the internet. There are a lot of them.
I mean, it kind of requires me to create another example with the scoring matrices and the phylogenetic tree.
No, the scoring matrix is already fixed. Just download it from anywhere and use it as it. You never change it. Just download, use it as it. You really don't need to change it. It's a standard. There are a lot of them, maybe 50.
Well, I didn't do that. Yeah, so I concluded that Mummer 4 demonstrated better performance, and then for the future directions, they can refine the core algorithms to improve them because GS-Align didn't really show what I expected to see. And then another direction they can take is to make some standardization for the input and output, because every tool has its separate input and output format, and that kind of makes it challenging to analyze them.
That's an excellent point.
Yeah. Yeah, so these are the references.
Did you compare with?
Yeah, I mean, I run the same example with blast first. And
I didn't see it in the table.
No, it wasn't in the table. It was in the.
So this is how you figure out.
I mean, I put the 21 errors
Yeah, it would be
manually.
better if you have it.
Yeah. Yeah, basically that's it. Oh. Thank you.
So, question. You have about 5, 7.
Probably because it tried to align the mismatching region. Because I took the region that completely mismatches, and then probably it tried too hard to align it and just made a lot of these nips.
What does it stand for?
GS? Oh, I need to look into the paper.
But you read the paper, right?
Yeah, I did.
Yeah, maybe it could be related to local versus global. Some of them try to align towards sequence rather than the entire. That's a very interesting question. If you look into these alignments,
What do you mean one by one?
so check like source. Is it really 200? Because it tells you the alignment, how it looks like.
Yeah, but the thing is that output format doesn't show me like the alignment, the characters themselves. It shows only the coordinates.
I see. Yeah, you might want to extract this based on coordinates, like to see the alignment.
Yeah.
Or what kind of alignment, how they come up with that number. They could be using this heuristic to align rather than use sophisticated sequence alignment algorithm.
Yeah.
Out of curiosity, did you try to align a genome to itself?
No, but that sounds kind of too trivial.
I saw one paper, it's really interesting, that shows even if you align the genome to itself, some of the tools like show 80%. It means only 80% of the genome similar to the same thing, same genome. So it's weird, trivial, yes, but it's amazing how those tools deal with the genome. Very trivial problem should be like standard.
I mean, I gave them credit for.
Any question?
So you mentioned that when you were evaluating the you didn't look at the runtime, you were mainly looking at the matches.
Yeah.
Correct?
Yeah.
But did you notice any difference in the runtime? Like is there one that took extremely long or
No, they're all like instant.
Very quick. How large is the genome?
So
Bacteria. You have it.
no, this is this is the like 1700 and 1701 for the artificial example, and for the real example, it's like 1,600,000.
What is the execution time ratio between the scaling with the length?
No, it's all very fast. It doesn't change.
Are you running it ever?
No, on my own Mac, because it's not like hard computations.